Our next session is on viral infections and host genomics. Viral infections and host genomics, you don't want to miss this. There you go. All right, good.
Well, now everybody outside will think they're missing something.
That's
really
right.
Exciting.
That's right. So our moderators are Gail Jarvik and Casey Overby Taylor. So Gail and Casey, please take it away.
All right, well, we'll just jump right in with our first speaker, who's Andrea, and Tales from the Genome.
Thank you. Can you hear me? Okay. Thanks very much for this wonderful meeting and this invitation. So what I will tell you is a little bit of how we've been using genomic medicine to elucidate the host determinants of severe human papillomavirus diseases and try to infer novel therapeutic paradigms for management of those diseases. And I thank everyone that already mentioned some of those points. Very quickly, what I refer to with HPV disease is a pretty broad spectrum of clinical manifestation, usually starting with benign lesions involving any cutaneous or mucosal surface, usually skin warts or verrucae on the skins, or papilloma and condyloma when they're on the mucosal surfaces. If they persist, they can evolve with some different degree of dysplasia and eventually evolve in frank squamous cell carcinoma. Despite pretty much everything we know about primary and secondary prevention and treatment, we still have that HPV diseases cancers are 5% of the whole number of cancers diagnosed worldwide and 30% of all the infections-related cancers. And this is illustrating very quickly what is the natural history of an HPV infection. The incidental infection occurred through microabrasion in the epithelium, infect the basal layers. The infection can be cleared or can persist, evolve again in dysplasias, and cancers occur when virus is going to be integrating in the host genome, overexpress the virally encoded oncogenes and cause transformations. And the point of this slide is that there is essentially specific host functions that are going to control the fate of each of those transition. They can clear the initial infection, they can clear the dysplasia-induced regression of it, and I'm going to show you that can even clear cancers. And the work we do is essentially focusing on identifying those immunological checkpoints to correct them whenever we can or leverage them for therapeutic use. So this natural history at the individual level is reflected at the population level. So if there is a given population exposed to HPV, there's going to be a number of individuals that are going to have benign infections, dysplasia, and cancers. And of course, they're going to follow more or less the natural history throughout age. We're going to be focusing in the program we use. We focus on a phenotype first approach in which patients with very severe disease with refractory warts or aggressive dysplasia or cancers in earlier age are sequenced and worked up immunologically. And this is the type of work that we and others have conducted and led to identify a number of specific functions associated to control of HPVs. Many are residing in intrinsic antiviral responses in the keratinocytes or DNA repair. Some have specific myeloid function and antigen presentation involved. And some others in effector function of T and NK cells. All of those somewhat pleasing in function of the adaptive or innate immune system. So pretty broad type of functions that are necessary to control those viruses. And I'm going to focus on three sort of paradigm of genomic medicine applied to HPV infections, IL-2RG, ZAP70, and IL-7. Thanks again for Jennifer for introducing this fascinating topic, but this is a patient, one of ours, with a nasal squamous cell carcinoma associated to HPV-16, one of the more oncogenic one, small skin warts, sequenced and found to have an heterozygous mutation in IL-2RG. Again, key gene for development of the immune system. I'm not going to enter into this for this audience, but a gene that is placed, as Jennifer was pointing out, on the X chromosome. This individual had an heterozygous variant, and the reason why that happened is because the variant was present in the germline. The CD34 stem cells had the mutated allele, but a reversion to the wild-type allele occurred in a common lymphoid precursor, leading to complete reversion and normal T-cell development, while the NK cells retained the mutated allele and they were defective in their cytotoxic activity. So this is a patient that, for the instability of the somatic reversion, as well as for the ongoing cancer and the defect in NK cells, was transplanted. He underwent complete regression and cure at this point of his nasal squamous cell carcinoma, as well as his skin manifestations. This is how his NK cells look like. We talked earlier this morning about functional validation. This is an assay that looked at how the cytotoxic function of NK looks like, was completely flat in these patients before transplant, due to also poor expression of perforating granzyme in those NK. Upon transplantation, brisk recovery of NK cytotoxic function and granzyme and perforating expression, and resolution of that phenotypic defect, which resulted also in complete resolution of what was the imbalance in the skin microbiome of this individual. We alluded to the importance of metagenomic sequencing. This is how it looks, the skin distribution of viruses and bacteria in a healthy individual. 80% of your sequence are bacterial sequence in green. The darker green are actually viruses. And those are all HPV viruses in the different type of HPVs, alpha, beta, and gamma. This is how this patient looks like before transplant. The majority, or almost 90% of all his reads were viral reads of alpha and beta HPVs, and upon transplantation, this was restored in the normal bacterial viral distribution, as you can see. So a case that somewhat illustrates the importance of IL-2RG, but the importance of actually NK function to control HPV diseases. And another example of the HPV genomic medicine and the relevance of TCR signaling for controlling of these particular viruses offered by this other case in which a ZAP70 variants occurred. This is a patient that was actually originally reported by Ivan a few years ago. This patient eventually, more recently, developed a very aggressive squamous cell carcinoma in her forehead, refractory to any surgical and medical intervention. She had some skin warts and some small oral condyloma, and she had a compounded heterozygous variant in ZAP70, a gene, an adapter that is absolutely important for transducing the signal of TCR and leading to T-cell activation. This patient, cancer, I'm not sure if you project well, but that's how the cancer looked like. Squamous cell carcinoma looks like a nest of cells that have clearly an invasive behavior approaching the muscular layers rather than staying on top of the skin, embolizing the vessels. Those nests that you see in the vessel should be empty and having red blood cells, not cancer cells, and perineural invasion. So this was a cancer with very high-risk metastatic spread behaviors. And this cancer, one sequence shows the presence of high level of transcription of all genes of HPV beta 19. This is notoriously not oncogenic virus, but under the lack of pressure from the immune system end up generating fusion transcript in at least 5 sites, so integrating in the host genome and overexpressing all the oncogenes of HPV, especially classic E6 and E7 that lead to P53 inactivation and squamous cell carcinoma formation. So this is, so far, is the first case of beta HPV integration and cancers caused by these viruses. And this is again a patient that with bone marrow transplantation underwent complete resolution of her cancer and complete resolution of all the other skin manifestations. And I'll conclude with one more novel combined immunodeficiency. IL-7 deficiency has been recently reported by a large group of investigators. There is so far 6 patients that have been diagnosed with this combined immunodeficiency. All of them have some level of, pretty aggressive HPV skin disease, evolving to squamous cell carcinoma and other more typical opportunistic infections. IL-7 is incredibly important for T-cell development in the thymus, but it's also necessary for the homeostatic maintenance in the blood and in the periphery of T-cells. And these patients, this is a normal single-cell RNA-seq of a healthy individual. This is how one of those patients looks like. Complete lack of CD4 T-cell subset and mates, and the lack of lymphoid precursors in the lymphoid cells in the skin. So there is completely devoid of those resident memory T-cells. This is a condition in which it's tempting to think to substitute what is lacking, and so adding IL-7 in culture lead to, of course, proliferation of the T cells and anti-apoptotic effect of the cells of these IL-7 deficient patient, as expected in a healthy individual. And so it was tempting to think to develop this in an in vivo approach, and this is the molecule we're currently clinically developing. This is a human recombinant IL-7 fused to an Fc neonatal receptor that maintain a long-acting function. This is a dose-escalating protocol in which actually the first patient in this protocol was dosed in the clinical center yesterday. So we hope that this will lead us to success in the treatment of those diseases. And I'm going to conclude with this sort of summary slide, emphasizing the point that genomic medicine is important to understand and solve that case, as we were saying before. But the thing that is most important for me is what it tells me in terms of managing that patient. It's not only understanding with gene, but it's what I have to do for management. And of course, bone marrow transplantation is good when we know what gene and we know what type of platform, and if we have a safe platform to transplant it. But there may be other interventions that are useful for those diseases. IL-7 could be one. Checkpoint inhibitors have been already approved for squamous cell carcinomas. Therapeutic vaccination for HPV is being developed. There is, of course, ways to potentiate the intrinsic antiviral effect of keratinocytes. TLR agonist and imiquimod is the classic, but there is many others that are developing, and it's important for us to know what a patient may have and what may benefit with. CXCR4 blockade for WHIMS was approved a few weeks ago, the first oral blockers that could be useful for things beyond CXCR4 gain of function and WHIMS, and of course interventions that are emphasizing effective function of NK and T cells, so monalizumab, IL-15, adoptive T cell transfers. So that's the armamentarium that has been sort of built probably in the last, I would say in the last 5 years, thanks to genomic medicine approaches. And just my thanks to the clinical center and all the groups and collaborators that have helped us in this work.
Thank you. And we have Jean next.
Hi, everybody. Thank you so much for the invitation. And today I will tell you about the work that we have done to understand the role of CCR5 in the context of flavivirus infections. Okay, so CCR5 is a chemokine receptor, and its main role is in leukocyte migration. You can find this receptor expressed on a wide range of leukocytes, primarily a subset of macrophages and K cells and T cells. And leukocyte migration is initiated by binding of its cognate ligands, CCL3, CCL4, and CCL5. CCR5 deficiency is rather common. There is a 32 base pair deletion mutation that is found in the coding region that ultimately results in a truncated protein that basically never makes it to the cell surface. So people that have this variant don't have expression off of this allele. It's pretty common. The allele frequency is between 10 to 16% in individuals of European descent, and this is found in homozygous frequency at about 1 to 2%. And we know the role of this in the context of HIV. HIV uses CCR5 as a co-receptor for entry, and the importance of this receptor in humans was demonstrated many decades ago by looking at individuals that are CCR5 deficient. These are people that have 2 copies of the Delta 32 mutation, the homozygotes. In a random blood donor group of people, the frequency is about 1%, but in individuals that are HIV exposed but remained uninfected, the frequency is increased to almost 5%. Conversely, when you look at people that are HIV positive, you basically can't find CCR5 Delta 32 homozygotes. So in the context, as we heard this morning in the introduction, sometimes these benefits come at the cost of making you more susceptible to something else, and that is actually the case for CCR5 Delta 32 homozygotes. So I'll tell you a little bit about what we learned about how being CCR5 deficient may make you at higher risk for Flavivirus infections. So Flaviviruses are a group of pathogens that you can find globally distributed on the top, and these are all viruses that are transmitted by vectors, mosquitoes and ticks. And as you can see, dengue virus, yellow fever, Zika virus, West Nile, Japanese encephalitis, and tick-borne encephalitis are distributed basically all around the globe. They cause millions of infections annually, lots of morbidity, lots of mortality, and there are no therapeutics for any of these at the moment. So I will tell you a little bit more about what we know about West Nile, because West Nile virus is the primary flavivirus that circulates in the United States. So when you look at the outcomes of infection, about 80% of individuals remain asymptomatic or subclinical, and about 20% will develop symptomatic disease. These symptoms can be really severe, oftentimes leading to hospitalization, so West Nile fever, as well as the more aggressive neuroinvasive disease, meningitis and encephalitis. And the mortality rate among the people that develop symptomatic disease is about 5%. So we started getting interested in CCR5 primarily because we found that CCR5 was really important in mice. And so when you infect mice with wild-type mice with West Nile, you see about a 30% mortality rate, but in CCR5 knockout mice, you see 100%. And so this gave us evidence and reason to want to go forward and to see if this was true also in human beings. And so what we did was we were able to get some samples from West Nile virus infected individuals that ended up on the extreme end of this spectrum, these people that had severe disease, symptomatic disease. And what we did was we looked for CCR5 Delta 32 homozygosity and compared to random blood donors, where again it's about 1%, we found that this was increased to almost 5% in individuals that were West Nile virus positive and had severe disease. We then wanted to look at the other end of the spectrum, people that were West Nile virus infected but remained subclinical or asymptomatic. And these samples are very hard to get because they do not come to clinical attention. And so we worked with the American Red Cross that screens for West Nile virus in their blood supply, and we were able to identify people that were asymptomatic or had very few symptoms. And what we found was measuring the number of symptoms, we found that people that were asymptomatic and reported 0 symptoms, basically the frequency of Delta 32 homozygosity was 0, as opposed to the frequency found in people that reported 1 or more symptoms, which was elevated. And when we looked at the people that had symptoms in greater granularity, what you can see is that the frequency of Delta 32 homozygosity increases with increasing reporting of symptoms. So what about people that have one copy of this mutant allele, CCR5 delta 32 heterozygotes? And so what we did was we also looked at this population, and what we found was that they basically phenocopy wild type. So they do not have a greater number of symptoms compared, like we observe if you have 2 copies of delta 32, and they look identical to wild types. And this has also been something that we showed in mice. We basically had wild type, knockout, or CCR5 heterozygous mice, and found that basically if you have half the amount of CCR5 expression on your cells, that is enough to give you a wild type phenotype. So basically, you need to really have no CCR5, at least that's what this data shows, to be at increased risk for flavivirus infection. So what about other flavivirus infections? So, we and others have looked both in mouse studies and in human studies, and found some studies have found protective role for tick-borne encephalitis, but there are of note studies that have shown no association, and overwhelmingly the mouse data supports the role of CCR5 for a wide range of neurotropic flaviviruses. And I want to also note that the delta 32 allele frequency is very low in populations in where some of these flaviviruses circulate, such as in Asia, where JEV, Japanese encephalitis virus, circulates. And many of the other flaviviruses like dengue, yellow fever, they just circulate in places where we wouldn't be able to test for CCR5 delta 32. And so how does it actually work? Well, I think we know from our studies as well as many other studies that it is basically leukocyte trafficking, which is the function of this receptor. You can see here that if you don't have CCR5 in mice, you do have the inability to clear virus from the brain, which is where the virus ultimately ends up. And what you can see here, if you look at the brain sections, in red are circled leukocytic infiltrates. So you can see in the wild-type mice, a lot of cells traffic into the brain, and these leukocytic infiltrates tend to co-localize with viral antigen. And in the absence of CCR5, you have just a lot fewer cells that get into the brain. So it seems like CCR5 deficiency just impairs the ability of effector cells to get to where they're needed to clear the virus. So as many of you may remember, in 2015 and 2016, a new flavivirus, not a new flavivirus, but a Zika virus, which is reemerged and caused a multi-country outbreak. And Zika virus is very much like all the other flaviviruses, but it has a couple of new features, which is that it has been shown to be sexually transmitted because you can find the virus in seminal fluid, but also the virus can transmit from mother to fetus. So if the infection happens while you're pregnant, the fetus is at risk. And these are two new features that are not known to happen with any of the other flaviviruses. And so we really are interested in this. The spectrum of outcomes for Zika virus and maternal infections is as great. So 70 to 80% of individuals or maternal infections result in no transmission of the virus to the fetus, but 20 to 30% do, and among these you also see a wide range of outcomes, fetal loss, as well as congenital Zika syndrome, which includes microcephaly, but then you do see that most babies are born healthy, but as we've tracked some of these babies, it has been shown that a subset of these do have some neurocognitive abnormalities as we track them into early development. So this has sort of made us question or think about whether there's a role for CCR5 in Zika virus infection, and we have preliminary data that sort of suggests that that's true, and it's really, I'm not going to share that data today, but it's made us really think a little bit more broadly about what the role of CCR5 might be, because we've always thought about this as really controlling cells migrating into the brain, which is protected by the blood-brain barrier, but it's possible that the role of CCR5 could be a little bit broader and control things, other barrier-protected tissues that are protected by the blood testes barrier or by the placental barrier. And so things to consider, we don't know how much CCR5 is required to give you protection. We know that 50% or about half the expression is enough, but how much loss of CCR5 do you really need to make you more susceptible? We don't really know that. And this is important because CCR5 delta 32 isn't the only mutation. In CCR5, there are other mutations that can control the amount of CCR5 density on a cell. And of course, you may know that there is an FDA-approved CCR5 inhibitor called Maraviroc that is on the market, and as well as several in the pipeline, monoclonal antibodies, which are all are being considered for chronic use. So if CCR5 blockers can mimic a CCR5 delta 32 genetic mutation, how much would that put you at risk for increased susceptibility to some of these flaviviruses? And of course, we would love to be able to know if other flaviviruses are affected, or the loss of CCR5 would put you at risk for other flaviviruses as well. And as I mentioned with Zika, it's made us really think a little bit broader about how CCR5 is protecting. CCR5, knowing your CCR5 delta 32 status, I think, is important, not only just for HIV, but also because it could put you at higher risk for neurotropic flaviviruses like West Nile and TB, where these genetic studies have been done, and could be broader than that and could include other flaviviruses as well. We just don't know. Of course, like I mentioned, that there might be a possibility that the loss of CCR5 could also put you at higher risk for poor pregnancy outcomes for flaviviruses that do impact pregnancy. And you can learn your CCR5 delta 32 status by commercially available genetic testing. With that, I will end. I just want to also say one more thing, is that this is pretty important, particularly even if we live in a country where West Nile is the only flavivirus that circulates. This is also important for people that are living in endemic regions where flaviviruses are, which is basically all around the globe. And this is important for travelers as well. And individuals that are at increased age and are immunosuppressed to some degree, this might be very important to know. And with that, I will end by thanking the people in my lab, but also most of this work was done while I was here at the NIH and Phil Murphy's group. I'd also like to thank the American Red Cross and state departments of health that provided samples for us to be able to do these studies. Thank you so much.
Thank you. Moving on to Priya.
Okay. Can you hear me? So make sure. Okay, thanks. So I'm going to be talking to you today. First of all, thank you for including me. My career has always saddled somewhere between genetics, NHGRI, and NIAID, and so I'm going to speak to you today with both hats on, but talking to you more about population level work versus individual. And we're going to talk about it through this idea of viral infections, specifically hepatitis C. So just as a reminder, I don't think this group needs to hear it, but I think it's important to be reminded that we're not only interested in treating those at risk of disease, but also in preventing and disrupting transmission, because that's really important on the larger scale of infectious diseases. And the underlying presumption that a lot of people have, not necessarily people in this room, is that everyone who's exposed will be infected and everyone infected will have a similar outcome. And this is why so much of infectious diseases has centered on important population measures. That means clean water, sanitation, and vaccines, which are phenomenal. But I do think that things are a little bit different than what we perceive them to be. And that's that some never get infected despite being exposed, and those exposed don't always have the same outcome. So there's something that's driving that heterogeneity, and we believe, and that's why we're gathered here today, that it's likely genetic. So there are really good examples, I think, that we can think about. The best really is the Cutter incident with poliovirus, where you had activated polio vaccine that was accidentally administered instead of inactivated to school-aged children. And in these 200,000 children that were injected, 40,000 had infectious symptoms, 200 ended up with paralytic polio, and 10 died. So this is where you have the same inoculum, you have the same age child, and so you've restricted your variables, and yet you still see differences in what what those outcomes are. And then we all have lived through a pandemic, and we know that in the early days before vaccine with the same variant, we saw multiple different outcomes in individuals from death to asymptomatic infection. And so what is driving that heterogeneity? Clearly factors in the pathogen themselves, and on a global scale, that's really important. So the dose that someone's getting, how that's transmitted to them, the variant, repeated exposures starting at a young age, and then host factors, obvious ones, age, sex, comorbidities, and co-infection. But I'm going to walk you through this example with hepatitis C virus, where an individual could be exposed to hepatitis C. Most commonly today, that would be through a blood-borne mechanism, usually injection drug use, but it could have also been from blood transfusions. And the virus would incubate for about 50 days. On average, you'd have some symptoms of malaise or jaundice or dark urine. This occurs in about 20 to 30% of individuals. And in the United States, that's about 19,000 new infections per year. And then that can lead to HCV infection, and then once you're infected with HCV, you actually have two paths, one of which is spontaneous resolution, and 15 to 20% of people will just spontaneously resolve their HCV infection. And the rest will go on to a chronic state of persistent infection, where factors like age, race, HIV co-infection play a role in that. And then if you are chronically or persistently infected, you can go on to cirrhosis and then hepatocellular carcinoma or liver failure. So we're most interested in that first level of who's resolving infection. So it's sort of taking the opposite, not who's most likely to have a severe infection, but why is it that some individuals naturally resolve that infection? So before I can get there, I just have to remind everyone, and Jennifer did it earlier in her talk, but there's one thing that makes infectious diseases just really different, and this is probably most aimed at the geneticists in the room, and that's that we have to deal with exposure, right? So it's not just about exposure in the cases where you presume there has been exposure, but also in the controls. So did everyone have the same opportunity to be infected and to have disease? And then how do you define that disease or infection, both in the cases and the controls? This seems really simple, but in the age of biobanks, it's really easy to just take access or get access to large numbers of controls and use them. And so I had a doctoral student who worked on this particular question of what would happen if you use population-based controls in this simulation instead of using well-characterized controls. So the yellow boxes are the well-characterized controls, and we'll start right at that very top box on the left, and the red is population-based controls. When the presumed prevalence of exposure is 100%, that means, you know, measles walked into this room and we all got exposed. There's no difference between the well-characterized controls and the population-based controls. But as you move down that panel on the left, it goes from 100% exposure to 75 to 50 to 25 to 5%, you get this inflation of your test statistic. That red box is moving to the right? And so we're now seeing things that shouldn't exist. We start to find genes that really aren't real. We start to see things that are complete, basically, we start to see spurious associations. And so if we look to the right, the one on the left was 20,000 cases and 20,000 controls. The argument has always been just increase the number of controls you have and you will control for it, but actually it stays all the way. And this has implications for how we look at population level genetics and infectious disease work. And so that brings us back to hepatitis C and why it's a really good example of where you can clearly identify your cases and your controls. So in this case, our marker of exposure is HCV antibodies. That's detectable throughout your life. It doesn't distinguish between chronic or resolved infection. It just tells me that you've been exposed to it, and that's that blue line on the right in the graph. Versus acute and persistent infection, where you would see HCV RNA, at least in the first 6 months, and then after that it becomes undetectable unless you have persistent infection, in which case you persist, and that's that purple line that you see that exists overall. So, I'm sorry, that green line that exists overall. So now I can see that you could have spontaneous resolution, I know you've been exposed, versus individuals who have persistent infection. And then we conducted a genome-wide association study. So this is a Manhattan plot looking at the minus log p-value on the left and then the chromosomes along the bottom. And we find, this really huge, large effect that is well known and well studied of lambda interferon. It's IFNL3 and IFNL4 on the end. HLA, also a clear contender to have a role in hepatitis C infections, and we identified a new gene, GPR158, in chromosome 10. So the key with this is a meta-analysis of of a large consortium of data that comes from around the world, acknowledging where HCV is actually occurring, both in Europe and the United States, and including African ancestry individuals. And we saw that when we stratified by individuals who were of African ancestry, that's the red, versus European ancestry, that's the blue, that when you increase the number of favorable alleles, that means your probability of spontaneously clearing infection, that your odds increased as appropriate, right? So these are independent effects, and it's dose response and related. And you see that in both ancestries that we look at. Now, individuals in the United States who are of who self-report as Black are more likely to have chronic hepatitis C infection, and yet we're showing genetically that if you carry these alleles and the allele frequencies differ, you actually clear at a really high rate. We also looked at sex-specific genome-wide associations because we know that there are sex-specific differences. This is a Miami plot with the top being females and the bottom being males. And we still see that lambda association and we see that HLA association, but we actually see in males this association that does not exist in females with this specific gene. So something that is specific to males overall. And then we evaluated both the lambda region with a lot of fine mapping in an effort to try to find the causal alleles, as have many, including several folks here at the NIH, and the MHC region. And we had more success with the MHC region where using trans ancestry meta-analysis, we were able to localize this region and identify binding HCV peptides and key amino acids that would play a role. So we took a really large region identified by GWAS and were able to identify specific peptides that were critical. And we believe this sets the stage for enhancement of HCV vaccine efforts. So in following Terry's instruction, outside of discovery, why do these findings matter? I think there's importance of the clinical translational aspect. Initially, post the original discovery of lambda interferon, it was immediately used in screening individuals because it predicted spontaneous resolution. And it was especially helpful when the therapy that was being administered was interferon therapy because you could see that those individuals were more likely to clear. It's also important for development or prevention, like I just mentioned with MHC. And then, of course, this helped to validate some of the findings we've seen that explain sex differences, ancestry, and other clinical and epi factors. But the real lessons, I think, for this group that we've learned from HCV is that population infectious diseases are just like any other complex disease. They're polygenic. There are multiple genes that underlie them. They're often independent. Our sample sizes in infectious diseases tend to be limited, but our effect sizes are really large, which means that we're really just finding the low-hanging fruit, and with more sample size and more information, we could find much more. Diverse populations matter. I didn't highlight it enough probably, but we only found a lot of what we did because of the cohorts that we utilized and because of the consortiums that we built that were international. And then, as I mentioned already, phenotype is key. This goes back to basic genetics, right? We need a strong and clear phenotype in both the cases and controls, or you can't find anything. And so you have to consider exposure. And this draws some questions on whether or not biobanks would be the most useful source of our time, or if it really needs to be existing or new cohorts. And so huge thanks to all of our team internationally that has worked on this. And I will pass this on.
Thank you, and Ryan.
Talofa lava, malole suifua lau lele, ile mamalu o e ua poto mai, a fifio mai, matala mai a ao. I'd like to thank everybody for inviting us to give a talk for me to represent the research that we're doing in the Pacific with Pacific peoples. And If I could just reorient us for a minute to this part of the world and center an often marginalized people. Pacific Islanders are rarely represented in research, and especially rarely in genetic research. And when they are represented, their data is usually aggregated with Asian individuals. Polynesia is located right here in the center, the Polynesian Triangle. There are 3.2 million Polynesians, or individuals who identify as Polynesian, living on the planet Earth. Many of those individuals live in the United States, New Zealand, or Australia. And then the largest of the peoples within the nation, the Polynesian nation, the Maori are the largest, followed by the Samoans at just over 500,000. Okay. My research team works with the communities in Samoa and American Samoa. Located right here, the Samoan Islands are divided into two nations. There's independent Samoa there on the left, and the US territory of American Samoa on the right. Okay. I represent the Olanga study group. The purpose of our study group is to try to sustainably and substantially reduce disease burden in the Pacific Islands, and especially as a model for ways to improve health in lower and middle income settings. Now, our research team is particularly focused on NCDs, so it's a little unusual for me to be at this meeting, but we had data that was relevant in an immune deficiency that appeared among the people of Samoa. Okay. My group has multiple cohorts that we've recruited from the islands, but the one I want to talk about in particular is Soifua Manuia, the Good Health Study. We have just over 3,000 genotyped individuals that were recruited in 2010. Okay. And of those, 1,300 were whole genome sequenced as part of the TopMed project about 8 years ago. And so this means that we have a resource that isn't really available anywhere else on the planet. We are custodians of a data set that gives the allele frequencies for variants that are present among Samoan people and could be inferred to be present among other Polynesian peoples. And so there are no known individuals of Polynesian or Pacific ancestry represented in NOMAD, despite its size. And so we often get a request from clinicians or from other researchers asking about allele frequencies so they can do investigations into variants of unknown significance that they see in patients or in research participants with Polynesian or Pacific ancestry. And so back when Twitter was a thing, a genetic counselor just put this out on Twitter and someone connected her to me, and then within the afternoon I was able to give her allele frequencies to help her with clinical interpretation. And then there's her update. Okay. We were contacted in 2021 by two clinicians in New Zealand who had a case of a child who died of severe viral disease shortly after vaccination with a live attenuated MMR vaccine. And it turns out that her brother had also died after vaccination a few years earlier. Subsequently, 4 additional kindreds were identified where a child had developed severe viral disease, 2 deaths, 2 that survived, and then 1 child who tolerated the vaccination quite well, but much later in life she developed severe viral infections. What's notable about all these kindreds from New Zealand is they're all of Pacific ancestry. Okay, the hypothesis was that these cases looked like they were individuals that had symptoms similar to known defects in the interferon-1 signaling pathway. And so the focus was to look at these 7 genes in whole exome sequencing. Now, there's a list of some of the very rare variants that are associated with immune disorders in this particular pathway to see if that's what's going on in these kindreds. So after whole genome sequencing, indeed, it was seen that the proband is homozygous for a truncation of the protein, and so was her brother, and her parents were both homozygous. And if you look across the rest of the kindreds, you see a similar pattern where homozygous individuals developed severe viral disease, and their unaffected relatives were all homozygous. Okay. And I'm going to skip through these slides pretty quickly. I don't think this audience really needs to walk through the science behind establishing it, but it expected to produce a truncated protein. That's demonstrated here with a positive control. It's not expressed at the cell surface. In the patients, MX1 induction fails when exposing patient 3 fibroblasts to type 1 interferons. And so in control patients, we see a response, but in patient 3, we do not, nor in the positive control. And then MX1 induction can be rescued with vectors overexpressing the wild type, but not in vectors overexpressing the mutant type, nor in the positive control. So what's demonstrated here is homozygosity for IFNAR1 and a change from the glutamate to a early stop codon is likely causal for these severe viral disease cases. And that's when our group, my team in particular, was brought in, was what is the prevalence of this variant? And the allele frequencies, this particular variant is present in Nomad. There's one allele out of 100 or 1.5 million alleles in version 4.1. But in Samoa, among 1,300 individuals, there were 32 alleles. We saw no homozygotes in our data set. And then in a data set from French Polynesia, about a 10-fold lower allele frequency was observed. Okay. What this means that among Samoans, we would anticipate that 1 in 41 individuals would be a heterozygous carrier, and 1 in 6,400 individuals would be homozygous and potentially affected. If we look across the Pacific, the size of the circles represents the sample size from which the estimate was collected. So in Samoa, we see a allele frequency of about 1.25%, and not too dissimilar among the Cook Island Maori. And the allele is also observed in a few other samples from French Polynesia and Fiji, but then is not observed in a few other samples, but many of which are small, so they might not be large enough to observe something with an allele frequency quite this small. Okay. For comparison, these are the population prevalences in the United States of several conditions on the recommended uniform screening panel: sickle cell disease, cystic fibrosis, congenital adrenal hyperplasia, MCAD deficiency, PKU, and X-linked adrenoleukodystrophy. And so among Samoans, the population frequency of this allele is high enough that screening might be something that you'd consider for this population or this community. Okay. And estimates would say that this is about the number of homozygotes that we would anticipate just among Samoans, and there's about 500,000 Samoans on planet Earth, just among Samoans, with some ranges depending in certain communities on whether they report being Samoan or of multiple ethnicities. Okay. As a reminder, Samoa had a severe measles epidemic in 2019. Which plays a role in our thinking about the phenotype. In October of 2019, the outbreak began. On November 20th, they began a nationwide vaccination campaign. They vaccinated 95% of the population in 7 weeks. At the time, this was considered unprecedented to be doing it at a national level. This was 2019. Such that the epidemic ended by late December. There were over 5,000 cases of measles and 83 deaths, most of whom were children under the age of 4. And there are circumstances why this particular epidemic occurred. But what was unusual was, if this population has 34 homozygotes for this particular allele, there wasn't a single adverse event recorded with the measles vaccine during the course of the campaign. So what does this say about the prevalence of this particular variant? It meant we were very cautious we were thinking about sort of publicizing this particular variant and publishing it in the literature. Okay. A couple of updates since the publication. Additional cases have been observed in New Zealand. Approximately half of those cases are now observed before MMR vaccination, so upon exposure to just wild yellow fever virus or chikungunya or RSV or influenza. When a Pacific individual presents with severe viral disease in New Zealand, such as severe bronchiolitis, they are tested for this variant. Originally, whole genome sequencing or targeting sequencing were the approach, but there is now a genetic test in New Zealand at $40 New Zealand dollars, about 23 US dollars. Okay, this is the ordering form. Okay. Regarding screening for this population, so New Zealand's approach. Okay, and to remind, New Zealand has 1.2 million Polynesian individuals. About 1 in 5 New Zealanders is of Polynesian ancestry, either Maori or Pacific. And this is also one of the few nations where I think indigenous peoples have significant roles in governance, in particular health governance. So they were incorporated into decisions about screening. So should we do universal screening? And the consensus was no. Community screening of Pacific people also know there were concerns about driving vaccine hesitancy. If we started screening all people for this variant, would people stop getting the vaccination for their children out of concern that they might be carriers? Screening all respiratory admissions, no. But what the nation has implemented is testing of specific individuals with particular presentations. Okay. And to date, no cascade screening has been done of the families, so there's really been no opportunity to try and locate sort of unaffected siblings. Okay. And to take this outside of the Pacific, we can rotate the world a little bit more and look at the Arctic. In the same issue of the Journal of Experimental Medicine, a variant was identified among the Inuit with similar patterns in some families, and similarly demonstration that it was located in the interferon-1 signaling pathway. Also rare and nomad, 2 out of 1.6 million alleles, but an allele frequency of 3.4% in the Inuit and 2.4% among Greenlandic individuals. So here we have 2 examples of genetic variants that are not extraordinarily rare, existing in certain populations which are generally underserved and marginalized. As a footnote, the original kindred the family has undergone in vitro fertilization, and through pre-implantation screening has recently had a baby boy who is wild type, and is quite healthy. The paper was published in 2022. I'd like to recognize in particular, we incorporated the feedback of 4 clinicians and advisors from Samoa, in particular in crafting the public health messaging associated around this paper. And then finally, I'd like to thank the Olonga Research Group for all their work and our funders. Thank you.
Right, thank you to all our speakers. We're open for questions, and I see one over there. Or is that a flag up? Can you read that? Yep. Okay, that's fair.
Thanks. That was an extraordinary session. I want to just ask one quick question of Ryan. It was quite impressive that in the measles population vaccination campaign, you didn't see any cases of severe disease. You know, in BCG vaccination, it's very clear that if you give BCG on day 1 of life, the risk of getting severe disease is high. If you give BCG at 3 months of life, the risk of getting disease is really quite low. Do you think that had any role? That is, in the aggressive campaign, were children getting vaccination at much later ages and therefore less likely to develop severe disease? Or was there a change in the lot of vaccine or something else?
I don't know, and I'm not a clinician. I don't know enough about the details relating to that, but I think one of our hypotheses was that measles killed the children who were susceptible. So the virus itself during the outbreak might have killed those children before they were subject to the vaccination campaign. So they might have been early deaths during the outbreak, in addition to adults who were affected. So that's sort of where we were coming from with it. But I don't know if enough information about whether or not lot number or anything like that has changed.
I see, is that George? Is that your?
Yeah, I just wanted to agree, really fascinating session, and a quick question for Priya and another for Ryan. Priya, when I listened to your session, I kept thinking of Jeffrey Rose and the classic paper on sick individuals and sick populations and the whole issue of resilience. And I didn't hear you mention anything about social and environmental influences when you have the same age group, race, gender, people in Chicago versus people in, say, New York City, where exposure may be the same, but you have completely different responses in terms of ability to clear. What do we know about that?
Yeah, so I think that ours was predominantly, so yes, perfect. I also think of Rose, and I think about the message that Jeffrey Rose makes about really focusing on populations, and genetics sort of turns that a little bit and says there can be personal aspects to this along with populations. In our case, we're looking at injection drug users, followed often in large cohorts that have similar protocols. So I think we may have eliminated some of the other aspects that you would have for other infectious diseases. But I do think those are especially important when you're comparing them. So we don't see big differences from that social environmental side in this particular scenario.
Maybe a very quick one for Ryan. The responses you showed to the questions about sequencing and those no responses, were they the responses of the community members or those of you experts working with them? I mean, I wasn't sure.
Yeah, I believe those are the decision that Te Watu Ora, the New Zealand Health Authority, made about whether to implement community screening, universal screening, community screening. So those were the no's and the yeses was their approach that the public health officials in New Zealand chose to take.
By the way, Eric Green is really supporting a meaningful community engagement approach to genomics and genomics medicine. We haven't started the effort yet. I'm sure Terry will be telling you all about it once we get started, but we would be really delighted to hear the community members' perspective as opposed to either the leadership or the scientific community. Really very, very important in community engagement efforts.
Hello, I was intrigued by Jean's presentation and your comment that that CCR5 mutations are actually on 23andMe. This may seem like a silly question, but what is the role of companies like 23andMe? And I mean, given the real desire for a lot of patients and individuals to control their own genetic testing and to have that information, I mean, can that play a part in a lot of the efforts that we're talking about in terms of increasing genomic medicine and testing?
That's a good question. Yeah, I think those commercially available testing platforms are useful. A lot of people use them for ancestry and things like that rather than trying to figure out what their, polymorphisms are. And so, but they do offer value. If you know what you're looking for, you can look and see whether you do have, for example, a mutation in any particular gene that's on their panel. Yeah, I do think that there is value in knowing that and having access to that. I think if you, particularly people that, know what they're looking for, can certainly search through their database and their own data to find that information.
Stephen and then Bob.
Question for Ryan. So I work at a children's hospital in San Diego, and as you probably know, sick babies in Samoa would go first to Kapiolani in Hawaii, and then they wind up at Redi. And so we do a lot of genomes of so-called Polynesians, and see a lot of this issue of how do I interpret this genome. It's really tricky. My question was, now that we're getting into genome-based newborn screening and we want to qualify a variant set for use in diverse ancestral populations, how much, genetic diversity? You showed one example, so first of all, for that one particular variant that you've done very nice studies on and have looked all across Polynesia, first question was, is your conclusion that within the Polynesian islander populations that there are significant differences in allele frequency or not?
I think the jury is still largely out on that. I think there are differences between different island groups, particularly the degree of admixture that you see between different island groups as well. There's some evidence that there is, in particular, a jump in allele frequencies from West Polynesia, so Tonga, Samoa, Tuvalu, Tokelau, and Niue, and then pretty much the rest of Polynesia, that's Eastern Polynesia, that there was the founder effect of jumping from that one set of islands to the other set of islands might have created some allele frequency differences there. But beyond that sort of hints at that, I think the folks in the Pacific, we haven't really developed a consortium yet. There are Pacific Islander and Polynesian geneticists who are now junior faculty starting to move up, where that's going to be a priority for us to do. I think the jury is still out on just to what degree we see founder effects that lead to allele frequencies between the different ethnicities within the region.
So just teeing on from that, have people measured fixation indices in these specific populations, and are they significantly different globally, or do they all fall under that, less than 0.1 difference?
Trying to figure out how to answer that question. I'm trying to think about how to not answer this in like a dichotomous way, like are they different or not, because there's really not an answer to that. I would say no other population on the planet serves as a good imputation panel for Polynesian individuals. So predicting allele frequencies from, say, East Asian individuals is abhorrent. So the degree to which, say, fixation indices indicate a significant divergence from the population, I don't know that anyone's done that study, but I'm going to say that they're high enough that we consider these populations dissimilar from a genetic level.
So Bob, and then Terry.
Thanks. I had a question for Dr. Lim. So it looks like populations that are exposed to flavivirus have lower CCR5 delta 32s, and it's protective against HIV in populations that have a higher frequency, such as in Europe. They're not as exposed to flavivirus. But prior to HIV, what do you think is responsible for the increased CCR5 delta 32 allele frequency in non-flavivirus exposed individuals? I remember reading something about, well, maybe it was plague, maybe it was bubonic plague, maybe it was this, maybe it was that. What do we actually know about that right now?
Yeah, that's a great question, and one that I spend a lot of time thinking about. There have been lots of theories about what was the pressure that sort of caused the enrichment, particularly in Europe, and there is definitely this decline that you see, particularly when you go towards Northern Europe. And I think the thing that the theory that I feel has the most weight is one where they talk about some sort of hemorrhagic viral plague that basically came multiple times over the course of multiple centuries, rather than one plague that happened and killed off 50% of Europeans would really not do the enrichment that you've seen. And so I think that they don't know what the pathogen was, but it was probably something where CCR5 was either very important. It was definitely has to be that the CCR5 was definitely important. So I would say that it was probably something, a viral hemorrhagic pathogen that hit Europe across multiple places for centuries.
Is it conceivable that it could be genetic drift in the absence of purified selection from flavivirus?
Yes, so there have been at least one very good study that was done that seems to that has proposed that is possible. But I would say that probably the field generally thinks that there must have been some sort of pressure.
Yeah, again, for Ryan, I'm a little curious when, it seems as though when you screen for this, you would, the treatment is just don't give them a vaccine, but you'd be screening for those that already have life-threatening infections. So what's the action then when you find it?
The first thing that comes to mind is make sure everyone around them is vaccinated. So herd immunity to protect the individuals, right? So it's interesting the contrast between by vaccination without ruling out an immune deficiency seems like, well, if we could do that, that would sort of perhaps improve, especially potentially vaccine uptake. On the other hand, the best way to protect these individuals is to vaccinate everybody in the community so the virus can't make it to them. Does that answer your question? That's sort of where my mind goes first.
Sort of, but not exactly. Because you know, obviously the kids that they're surrounded by would be the ones you'd want to vaccinate, but also if they're siblings, they'd be the ones most likely to also carry the variants. And so it just struck me as a little bit odd that you wouldn't test everyone to catch those, you know, asymptomatic ones.
And I think it's a matter of resources right now. In New Zealand, I think it's a matter of resources in terms of it's not risen to the level of being, I'd presume, a health priority to sort of do cascade screening or anything like that.
So one thing that we might want to think about in terms of future implementation or development is are there ways to develop really cheap tests for this at the point of care that might only be for these variants, for example? And is that something we should talk to our technology development program at, you know, within NHGRI or in other settings to develop? Is that something that you think might be worthwhile?
I don't know, because I don't know how soon newborn screening with a whole genome, or at least a whole exome, you know, like if that's coming sooner rather than later, do we need to invest in, you know, do we need to invest in technology and delivery systems for all these individual variants when we might be able to just capture it all?
Well, interesting point. On the other hand, if they can't afford a single variant test, how in the world are they going to afford a genome test? So I don't know, those of you who are in the technology field, and I know we have some folks here who have feet in both camps, you know, might want to comment on it. Or Jennifer, you just raised your sign. Maybe you want to comment specifically on that?
Yeah, well, one thing to mention quickly is New Zealand was the first country after the US to institute nationwide newborn screening for SCID. And so I think that was very forward-looking of them, and quite interesting that seems different from what you are describing, although I know that the Maori population is involved in their health decisions.
There's government administrations change and priorities change, and there's been a recent shift in New Zealand.
Yeah. And then another thing I did want to also mention is that there are places that have implemented TREC screening, which is obviously a PCR test, and have added in particular regions where there are variants of you know, significant incidence, they add these things that would not necessarily be detected by TRECs. And one of them is in Canada, in the Northwest Territories, where there's a frequent ZAP70 incidence, and ZAP70 is it's a T-cell immune deficiency, but it affects T cells after the stage of T-cell recombination and TRECs. And so they have added this particular variant to their, to the newborn screen to pick up those cases, and have done it successfully. And I think there are other places in the world that do that as a step, between here and way before whole genome sequencing.
This is a question also for Ryan. So I completely agree that under or non-represented populations need to be. We need the database information for what is common, what's not common, but. That could be a double-edged sword in the sense of, if you go back to that Twitter exchange you showed, right? You mentioned, this variant is present in 2% of, I think there's Maori population, so therefore it's a polymorphism, it can't be disease-causing, and yet you showed with the interferon alpha receptor variants, right? So both in your population and the Inuits, that those MAFs were, around 2 to 3%, and yet those are not polymorphisms. So, how are you, even though you have this population data for this small, essentially bottleneck population, how are you going to distinguish, what is a founder effect versus what is common?
We get things wrong. science, right? That's a really hard question. That's a question we're sort of always asking ourselves as we walk in. My mentor always told me, know your assumptions, right? Like, so when we deliver that genetic information about the allele frequencies, know that you're assuming that just because something appears to be polymorphic, you're assuming it's not going to be involved, but we could be wrong. And yet that's sort of the standard by which we are often looking at databases like Nomad, isn't it? Is, oh, this is common enough. You know, we don't think of European ancestry as being a founder population, you know, because of admixture, but, you know, and it's been, white people have been pretty well studied. So we don't think of it that way, but that is, you're right, it is a double-edged sword because we might have ruled this variant out if a priori it was known that it was common, this common.
So I'm going to call on Dusan in a minute, but I'm going to interject nomenclature, which people know I want to do, which is a polymorphism is not synonymous with benign, right? A polymorphism is 1% allele frequency or higher. It can be pathogenic, and we have many examples of that. So just a nomenclature point, but.
So to add to this discussion, and maybe to what George mentioned as well, when we are forming policy on return of genetic results that may impact infectious diseases here, or in this case, vaccination decisions, right? I think we have to discuss among ourselves as to what is the best way to do that. How do we include the vulnerable populations in that decision? How do we make sure that we do it in a way that it doesn't become a political fight, but it becomes a rational decision for the betterment of health of the population while protecting the vulnerable. And I don't have a solution for it. I just think it's something we constantly have to keep talking about, evaluating, because even the ethics of genome sharing and consent has been changing over the past 2 decades, and we have evolved with it. By no means we're doing it right now, nor we were 20 years ago, and I don't know what's going to be done there, but I think we have to keep talking about that at a lot of these things, so we don't end up in a situation that becomes a political choice as opposed to a rational medical cost, population benefit, and inclusion decision.
Let's see, David?
Yeah, so this may be more of a comment than a question, but thinking about implementation, and I think the concept of windows of opportunity is really important. As Terry kind of just brought up, this conversation around, do you do things that you could do now with a few number of variants when maybe not too long it'll be the whole genome. And I was also thinking about, we'll talk more about this tomorrow, when Priya was talking about interferon and the genetics of interferon, which was incredibly relevant very recently for hepatitis C, but now with incredibly effective therapies and nobody being treated with interferon and trying to move toward a single shot cure, the genetics may wane for that, and some of that also comes up with CCR5 and other things. So I'm not sure how much of the talk today has to do with, or talks have to do with windows of opportunity, because you're talking about fundamental genetics that affect susceptibility to an infection. But as we talk about interventions, whether that's a genetic test intervention or a therapeutic intervention, I think that's a really important thing in terms of thinking about how to prioritize with resources and all that. So not a question, but more of a comment.
Any other speakers want to respond to that comment? No disagreement, certainly. Bob, go ahead.
Thanks. This is also by way of a comment, which is, I think both pharmacogenetics and infectious diseases are really great examples of where allele frequency has a lot to do with exposure. I know Steve Holland has published this really nice work on severe cocci, where the allele frequency of the variant that confers susceptibility to severe cocci is actually high enough to make 25%, I think, of all Amish heterozygous for it, and yet there's no cocci in middle Pennsylvania. So it was ruled a benign variant for many years because of its high allele frequency, but it's clearly not when you look in the right population. I think we're going to see more and more of that. In fact, the whole idea of the current clinical classification system where a single allele frequency cutoff is used for recessives and dominants, like two different ones, but just one for recessives, one for dominants, probably is not subtle enough to really reflect the realities of whether variants are pathogenic or not.
Yeah, I completely agree, Bob, and I think that Jean's talk also speaks to the other aspect of infectious diseases, that selective pressures likely have created environments where you're going to have some benefit to some of these alleles and risk based on the other pathogen that you encounter. And I know in other research that we do, we see the same thing. We see some protection against infectious diseases, but it puts you at much greater risk for autoimmune disorders. So it's just balancing that, like which side are we looking at?
Dusan, did you have another question, or okay, any other questions? Otherwise, I have a question for Andrea, which is, you talk about HPV, remind me of the amazing success that vaccine has been to reduce these cancers, but we have increased vaccine, and we had another mention of vaccines as possible treatments, but there's this increased vaccine hesitancy now. And how does the medical community try and counter that, I guess is my question, or what are our options here?
Yeah, vaccine is incredibly effective. It's changing and shaping the circulation of the oncogenic variants. But the problem of some of the immunocompromised population is that in addition to the issue of vaccine hesitancy and in addition to the problem of actually getting the vaccine, the response to the vaccine in this population is not great. So we measure it in a population of T-cell lymphopenic individual, their antibody response to the Gardasil 9, it's greatly impaired. So whenever there's going to be a background of T-cell deficiency, we know that it's not going to be an effective one. And the other issues with HPVs is how, and we don't know yet, how is shaping and changing the population of oncogenic ones. There's 450 classified HPVs so far, so the oncogenic one, fortunately, are a minority, but it could be expected that could be a shift that is including non-oncogenic variants or not included in the current platforms. And the other thing is that in our immunocompromised population, you start having, or you have cancers like the case that I showed you, in which you're actually having cancer with integration of a beta HPVs, which is definitely not even on the radar. So far, cutaneous cancerogenesis has been attributed only to UV exposures, and the beta HPVs have never been implicated. The more we start looking at sequencing the tumors, and this was to go back to Jennifer's comment about sequencing in tissue beyond blood, the more we're actually seeing integration of variants that we didn't think that were important. So yes, definitely vaccine, but there's probably going to be more.
Any more comments?
Yeah, if I may. And we do have a fair amount of time, but we don't have to take it all. We can, we can stop and just finish a little bit earlier. But I am a little bit curious when it comes to particularly like the hepatitis B and C stories. Are there interactions between the host genome and the genome of the pathogen? Because there's been, I think, a lot of work done on sequencing of the pathogen, and David may talk about it with HIV, et cetera, tomorrow. But can you comment at all about how the specific variant you're infected with may affect, may interact with your own genome?
Yeah, thanks, Terry. That's exceptionally important and something that we've tried to do for quite a while. It was really hard with hepatitis C because we're using often historic samples to do the work that we do, because you've got to build the consortiums, follow individuals, know their outcomes. But we are doing that with hepatitis B. And so we're able, because hepatitis B in parts of Africa can occur at much younger ages, so we can get both the sample and the sample of the pathogen and the individual. But I do think that that is the complexity is, and we've seen it in viral diseases, but that complexity of the viral genome and the human genome having that interaction. And it could be really for any pathogen.
Well, because one of the things that comes to mind is cystic fibrosis, and as I understand it, in cystic fibrosis, each patient sort of gets their own Pseudomonas strain that kind of encodes on them and stays with them for years or life. And is that genomic, or is that just whichever one happened to establish itself first? And it's probably a little bit of both. And I don't know if there are cystic fibrosis Pseudomonas people around the table or not, but might be another sort of model of that.
Scott?
Yeah, that just got me thinking, because I'm a rheumatologist. Some of the biggest susceptibility factors for lupus and for MS are things like EBV, but the mechanisms of that are totally unknown. And so is there an opportunity within biobanks, like you kind of did for CMV, to look for some of the genetic characteristics or interactions between people who are reactivating things like EBV or even CMV and susceptibility loci for autoimmunity?
I do think that's where biobanks could be useful, is EBV, CMV, like pathogens where there is high prevalence and presumed that the entire population has been affected. I think you really can do a lot of work, and I think that's what we saw with MS and EBV, that work that came out of large-scale studies. So yes, I think that's one of the better benefits. And there could be other pathogens. You just have to think about what is circulating by a certain age in a large portion of the population.
It's Liliana again from this corner over here. We're just going to respond to some of your questions regarding pathogen adaptation to living inside a host. So many bacteria actually undergo very severe dramatic genomic reduction once they become fixed into a human host, especially once they're carried for a long time. We saw also in COVID, there's a specific idiosyncratic evolution of the pathogen inside the host and even inside different tissues. But whether those trajectories are the same, different human hosts, are questions that we're currently exploring through our genomic centers as well as our systems biology programs. I forgot what, I was gonna mention, we do know, for example, in commensal pathogens, they also are, commensal organisms in your microbiome. They are also sort of unique to each person, and I think that kind of source data is gonna give us a little bit of better understanding of if there are human genomic or other variables that do make and have an effect on what genotypes can exist of those bacteria in your gut. The issue with most of our bacterial studies or other pathogens, fungal, viral, is that they're very short duration, right? So we can't see that large scale genomic adaptation and selection for other pathogens. As you would, for example, so CF is a great case, but it's also cystic fibrosis, so it's not necessarily the right model to study human genome, pathogen genome interactions.
So I had a comment for the speakers around this being able to capture exposures. So we brought up the biobanks a little bit, and I'm wondering, since it's important to try to find people who may be resistant, for example, what kinds of surrogates for exposure might be relevant to better capture in biobanks, so that some of this work could, some of the research could be more effectively pursued. I don't know if that makes sense. So, for example, on the topic of exposures, for example, there are examples on finding controls who were exposed but didn't actually get the conditions, and so if we were able to know whether somebody was involved in drug use, or some kinds of surrogates that could say that somebody's more likely to be exposed, are there opportunities to better capture these like surrogates of exposure in biobanks? And has there been any work in that space?
Yeah, Casey, I think it's a really good question. I think that's the problem, is there isn't a surrogate of that exposure that we have, right? So we diagnose disease, biobanks can work for that. We diagnose infections, but we don't know if you've been exposed or not. And so without that surrogate, it makes biobanks really difficult to use, because the assumption then is that someone has been exposed, but if they haven't, then you're not comparing apples to apples. So that's where cohorts have been really beneficial, because you can follow people over time and sort of determine whether or not they've been exposed through frequent testing.
But correct me, and Jennifer actually has a question, but I'm going to jump in anyway. So my understanding is that the Ohio River Valley, almost everyone who's lived there for any length of time has been exposed to histoplasmosis. Wouldn't that be one of many kinds of markers for exposure? Or living in Arizona, you've probably been exposed to whatever it is in Arizona. Is it blasto? I don't remember. But at any rate, you know, and trying to, is it cocci? Okay, sorry. Yeah, this is why I'm an internist, not an infectious disease person. But at any rate, you know, maybe, you know, coming up with some of those kinds of surrogates would be a very helpful thing, at least for the research, and maybe as markers for the patients.
Yeah, absolutely. I think that, you know, you can make some assumptions based on what you think is circulating in a given area. It's just harder to use a biobank to do that, right? So if you think of like the UK biobank, we can't make the assumption that the people in that cohort have been, in that biobank have been exposed to certain things.
I mean,
I was just going to bring up how complicated the whole field of vaccinology is. And you know, we've been sort of talking about vaccines as if they're, you know, one is equivalent to another. And yet we know that, you know, the reason MMR are live vaccines is they don't work if you don't have live vaccines. They don't give lasting protection. And on the other hand, now we have a whole new technology with RNA vaccines. Maybe we should be looking at how we can make vaccines safer for everybody, because we know that a certain number of people are going to have variants that cause big problems when they get vaccines.
Hi, this is Inka from Nile again. I have a question, and I noted this earlier, but Liliana reminded me. So there was, I think it was Andrea who said that in his talk about treating the immunodeficiency by, I think it was transplant, and then looking at the microbiome, and the microbiome changed before and after treatment. So I'm wondering, how common is the microbiome looked at after transplantation or after changes in immunodeficiency, and if this is commonly done, and how the microbiome might change before and after, because that could also have implications for subsequent infections, I guess.
Thank you. Probably Liliana can answer and can add on. So what we did was probably refer to skin colonization by HPV. So that's pretty ubiquitous signature. All of us are harboring some type of HPVs, and so that is pretty stable on our skin. And on the type of immunodeficiency that we studied in Nile, we've been seeing a very strong signature to enrichment of those HPVs in, for example, DOK8, GETA2, IL2RG, or other type of immunodeficiency. So that is translated in a somewhat more stable change. I don't think there is enough granularity to look into what type of taxa are more or less expanded. Those data are coming through now that there is more metagenomic pipelines. And in the particular case that I showed you, there was enrichment of the alpha oncogenic HPVs that should not be on the skin, should essentially be populating and colonizing mostly the mucosal surfaces. So in immunodeficiency, you also shift the regular tropism that you would be expecting for these particular viruses. but I don't think we have enough data to be systematically characterizing different immunodeficiency and shift in bacterial species, for what I know. Although the skin, I think, going back to sequencing of the skin and tissues and other opportunities is an incredibly accessible source. If we're able to identify, and going back to the concept of exposure and signature of exposure, if we're able to identify also through the skin particular features and signature that can be complementing data that are going to be genomic data, especially at the metagenomic levels. I think the skin reflects your immunological status very well. It's populated by any immune cells that is there, and from what we've seen, the skin microbiome does reflect a lot of what your immunological condition is. But again, we haven't systematically done that. Julie Seger has done a fantastic job with ID Kong, but still, it's early. I don't know. Probably Liana want to add something.
And to that, I mean, we're hoping that the microvirome that the NIH Common Fund is starting will give us some additional answers, at least what the virome looks like in healthy human populations. So a good comparator, hopefully.
Yeah, and just building off of Jennifer's comment about making vaccines safer, I remember a time many years ago when the argument was made that every clinical trial of any drug should include genetic, at least genetic material stored and tested later. I would ask my NIAID colleagues, is that common in vaccine trials? Is DNA stored and examined in vaccine trials? Inko, maybe that's a question for you, I don't know.
So do you mean human?
Yes, yeah. So to look for, in case of adverse effects, then obviously one could do the GWAS or whatever studies. This is discovery research, so sorry, but I'm just asking that question.
I'm not an expert on this. I would think very restricted. I mean, of course, and we know that some people respond better to vaccines than others, and I think there was a study that I remember that related to the circadian rhythm, and you have a better vaccine response, I think, in the morning than in the evening. I'm not sure how detailed people actually look at human genomics and and see how they respond. I mean, I would think B cell, if you have B cell issues and others, then you would have less of a vaccine response. So I guess the common players would likely be good candidates to look at, and maybe somebody else has a better response than me.
So I just might suggest for the meeting that you and Lucas are planning, maybe that would be a question that Lucas
I mean,
wants to
I know
respond.
who I could ask potentially. This is Maggie over here. Sorry. From NIAID. I'm from the Autoimmunity and Mucosal Immunology branch and Division of Allergy Immunology and Transplantation. There is a large cohort for COVID that includes a lot of healthcare workers that had samples before they got COVID, and then after they either contracted the disease, had the vaccine. And so have they done that? I actually don't know because I'm not personally involved in that, but that might be one that's recent enough where maybe they do have that information or have the samples stored for that. I don't know.
Not like precisely, but I don't think it's routinely done in vaccine studies. I think you generally try and measure like actual, correlates of protection. Obviously I think you run up, particularly I think in larger clinical trials and sort of the economics of that. People really want to save costs, and sequencing is probably one of those costs that probably, but back to the earlier discussion about biobanks, one thought I had, and I'm sorry, this is also very into a bit of discovery. I remember in graduate school, there was a discussion we had in my group about looking at, understanding populations that have a large amount of data and samples biobanked and have been exposed to certain pathogens over time, and then comparing those to populations that have recently been exposed to that pathogen and hadn't been previously, and looking at maybe differences in outcomes. Dengue is one of those that comes to mind because I remember that there was a study back in the 90s of a dengue outbreak in Cuba where there was a big, with dengue, there are 4 serotypes, and if you get infected with a different serotype after being previously infected, you're predisposed to have more severe disease. And Cuba's an interesting population because you have a mixed European population and African population. And there was this big wave, I think dengue 3 in the island. People got mildly ill, no big issues. And there was a big secondary wave of disease about 2 years later, if I recall, from a different serotype, and that you really saw quite dramatic differences, in severe disease distribution, based on sort of people's ancestry. And in my lab, or one of the labs I rotated in, they had biobank samples from Nigeria over years and actually found that dengue and Zika had been circulating in those populations for a long time, but didn't really seem to observe severe outcomes. And so understand what populations have been exposed in the past, what populations might be newly exposed, might be a way to leverage those biobanks to actually have more power to detect those genes that might be involved in the variation of phenotypic clinical presentation.
Yeah, thanks. I did want to come into this exposure discussion with the All of Us research program, wondering, I'd love to hear more from this community about what kinds of exposures would be useful to incorporate into our data set. Just so you know, we are going to collect residential and occupational history over the lifespan of participants as best as we can, and have geocoding to a number of linkages and geocoding to a number of databases, including starting off with the Environmental Justice Index. We collect social determinants of health data, and there's a number of other data types that I'm hearing this community would think would be important to understand the manifestations of certain infectious diseases. So maybe it's during this meeting or afterwards. I welcome the opportunity to learn more about what we can do to enhance your ability to do the kind of research we're talking about here.
So I was going to mention it before when the databases were discussed about. So maybe something to think about that we can all incorporate in both cohort and maybe the biobanks, even the large-scale ones, is seromics. Because with FIP-seq now it's not that expensive to do it, and you can find what you're not looking for, right? Which is what we also want to know about. What is it that we are exposed to, but we don't know that we are not routinely testing for. So I think that can be a rich beginning, at least in the pilot program, that has that type of depth.
Can you repeat what the type of sequencing?
I mean, FIP-seq is one of them. There is a few different ones. Basically, it's a library that has an antigen to it, and then you basically catch the antibody. It's basically a high-throughput ELISA, if you wish, that's sequencing-based and decently inexpensive.
Yeah, that would at least show history and overall serology.
Okay, I think you're done. So super, thank you. We're about 5 minutes early, so why don't we save a little bit of time and come back at 3:20. So please be back at 3:20.